So, hello everybody, welcome back to another histology video. Today we'll be talking about the questions of 21 and 22, which is the mucus layer of the respiratory tract and also the structure and function of the larynx and trachea. So, the respiratory system is split into two parts, the conducting part and the respiratory part. The conducting part is basically where air is conducted, it's in its name. Air is humidified, warmed, and purified. So when air is warmed, the reason why this is important is because when air is warmed, it opts for optimal gas exchange. And logically, warmer air keeps moisture in the respiratory ways, and this basically prevents the lungs from drying out. Um, and it's consisting of the nasal cavities, the pharynx, the larynx, the trachea, the bronchi, the bronchioles, as well as the terminal bronchioles. And in the respiratory part, it's basically where gas exchange just occurs. And you can see it in the respiratory bronchioles, the alveolar ducts, and the alveoli. Now, the conducting part basically consists of the pseudostratified columnar ciliated with goblet cells, epithelium. And we also have 5 different cell types in it. So we have the ciliated, the goblet cells, the brush cells, the stem cells, and also the neuroendocrine cells, which we'll talk about more in the next slide. And it also contains the lamina propria mucosa. To put it simply, the lamina propria mucosa helps supply nutrients to the epithelium and plays a role in immune defense. As you can see in the slide, there are macrophages, plasma cells, and mast cells. So that would help in immune defense. It also contains elastic fibers. It also contains serous mucous glands, and it also contains MALT, which is mucosa-associated lymphoid tissue. This is very important because many professors ask you about this. And in the schema below, you can see what a pseudostratified columnar ciliated epithelium looks like. They're all like different sizes, and the nuclei are in different heights, but they're all attached with the basement membrane. So the nasal cavity, again, basically heats and moistens up inspired air and also cleans it, like for example, with the hairs of the nasal cavity. But we'll mainly talk about the parts of the nasal cavity. So we have nasal vestibule over here, which is inside the nostrils lined by skin, also known as stratified squamous epithelium. And number two, we have the respiratory region, which is the largest part, and it is lined by the respiratory mucosa, like we've seen before, which is the pseudostratified columnar epithelium that is ciliated. Um, and the olfactory region, which is the upper part of the nasal cavity, and it contains a specialized olfactory mucosa known as the olfactory epithelium. To put it simply, the olfactory epithelium basically covers the superior nasal conchae, as you can see in the last slide, on both sides, and it just basically helps detect smell via the cribriform plate of the ethmoid bone, so it connects it to the brain. Olfactory neurons are bipolar neurons, meaning that they have two extensions, and yeah, they just detect smells and they send that information to the brain. Now, the epithelium of the respiratory system, like we said again, is the pseudostratified. The cell types columnar, basal stem cells, and ciliated columnar cells, which is the most abundant. And the reason why it's the most abundant is that it basically ensures that the pathway is equipped to move and clear out trapped particles or mucus containing anything that we don't want more efficiently. Yeah, so it basically just ensures that it's free from unwanted particles and pathogens. Um, and we also have goblet cells and brush cells, as you can see in the schema on the right. Brush cells, they basically have small apical surfaces bearing like short or blunt macrovilli. We also have granular cells in it, and intraepithelial lymphocytes, and also dendritic cells. And it's also important to note again and again, they're all connected with the basement membrane. We also have the lamina propria mucosa. We explained before what it was. It's loose connective tissue. Again, it also comprises of the collagen and elastic fibers, and it's also highly vascularized. As you can see, the vein is located there. And just more about the epithelium. The ciliated columnary is the most abundant, like we said before, containing approximately 250 kinocelia, and they exhibit some sort of mucociliary defense mechanism that basically just moves mucus down towards the esophagus. And yes, goblet cells produced by the mucus, as we all know. And these are the apical portions that secrete these large secretory granules can be sustainable with Altium blue. Very important to know. The brush cells, however, only make up 3% of the respiratory system. Um, but they have something interesting about them, and it's the fact that they have a sensory function. This is due to the fact that they are close proximity with the nerve endings of the vagus and trigeminal. And if you were to think about the anatomy of it, it would make sense why it would be having that function. We also have small granular cells, and so these are dark granular cells, which secrete catecholamines, serotonin, and calcitonin, which are catecholamines, serotonin are biogenic amines, and calcitonin are peptide hormones. These are important in the functions of the respiratory system, which I'll talk about in the next slide. And they represent something known as the DNES, the diffuse neuroendocrine system. Very important to know. Many professors would want you to know this. And the basal cells, again, it's just for the regeneration of the epithelium cells, for example, the goblet cells or the ciliated cells, just something important to note. And yeah, so here is just what it would look like in the electron microscope. And on the right, you can see a schema of what I just explained on the last slide. And it's also very, very, very important to note that the neuroendocrine cells are also known as the Kulczycki cells. Many professors want you to know this. Um, and I'll explain the function of it. So they are for the changing of the lumen, like the diameter of the lumen of like the respiratory ways or like the lumen of the bronchi, for example. And the reason why this is important because like when you inhale, for example, cold air, um, it will trigger bronchoconstriction, and this is because, to put it simply, it's to conserve heat and moisture. So it basically prevents the damage of the respiratory system in that sense. Next, we have the pharynx. We're still in the upper respiratory tract, but it mainly just connects the nasal cavities and the oral cavities towards the larynx and the esophagus, and it's a passageway for air and food and helps in speech resonance. We're still in the upper respiratory tract. And going more downwards, we have the epiglottis, which is just the entrance towards the larynx, which is attached towards the hyoid bone, sorry. And it contains two parts. We have the lingual surface and the laryngeal surface. So the lingual surface, which is the apical part, contains the stratified squamous non-keratinized epithelium, and the laryngeal surface contains the pseudostratified columnar ciliated epithelium with goblet cells. And we can also see that it contains most of it, the elastic cartilage, helps it in its elastic nature. For example, when you're digesting food, it needs to move. And it also contains the serous mucous glands, which contains both the mucous tubules, the serous acini, um, also equipped with the serous demilunes, which is filled with actin filaments to basically push the contents out from these glands. And mostly in the laryngeal surface, we have the lymph nodes. And like with other lymphoid tissues, like the tonsils and the adenoids, they play a very important in immune defense or immune surveillance of the upper respiratory tract. Yes, just to move on. Here we have just two slides of what we just talked before. So over here we have the mucous tubules and the serous demilune. And on the right, we have the elastic cartilage of the epiglottis, and you'll learn more about it in the previous video, which you should see just for reference. Now we'll talk about the larynx. To put it simply, it's split into three parts: the false vocal cords, the laryngeal ventricles, and the true vocal cords. Now, the false vocal cords and the laryngeal surface, they both contain the same thing, same contents. They are all pseudostratified columnar ciliated epithelium with goblet cells, and they all contain serous mucous glands. The only difference is that in the true vocal cords, it's very different because it contains a whole different epithelium, which is the stratified squamous non-keratinized epithelium, and it does not contain any glands. And it also has a ligament, which is elastic, and it also has a muscle, a striated muscle, which helps controlling the abduction and the adduction of the vocal cords for us to speak properly. Now, let me tell you the reason why the organization of the epithelium makes sense for the function of the vocal cords. So, like the true vocal fold epithelium basically helps in like withstanding like force and friction. And we can also see that it's highlighted as well in the fact that it does not have any serious mucus glands. Um, and that would make sense because that would get in the way of the force of vibration in our vocal cords. Um, and that is basically it. It also contains some large cartilage, which is the hyaline, and a small cartilage, which is the elastic. Here is just another schema. Here's just another slide, sorry, another slide of the larynx. You can see that the serous mucous glands are located in the false vocal cords and the ventricles. Um, and you can also see the ligament of the vocal cords. And yes, it's stained in green trichrome. Here's just a closer view of the ventricular fold of the larynx. You can see the mucus, the serous mucous glands, and stained in HE. Here is the ventricle, a closer view. You can also see that there is lymphoid infiltration, which help it in immune defense of the upper respiratory tract. And we can also see in the vocal cord of the larynx that we have the elastic ligament and the stratified squamous epithelium, which is non-keratinized. And you can also see over here, just a tiny bit, the muscle, which help us in opening our vocal cords. Next, we'll talk about the trachea. Now, the trachea is split into different parts. So, tunica mucosa, part one, tunica submucosa, part two, the hyaline cartilage, part three, which is the most like prominent, you can see, and the tunica adventitia. So, to put it simply, the trachea is basically a short, flexible air tube before it splits into the two bronchioles, the primary bronchi. It's having like a diameter of 2.5 centimeters and it's about 10 centimeters long. And its tunica mucosa is basically pseudostratified columnar ciliated epithelium with goblet cells, contains lamina propria mucosa. We explained the function of that before. And I forgot, sorry, we also have the elastic fiber layer, which is very important because it helps it in its elasticity. And we also have the tunica submucosa, which contains loose connective tissue and also the serous mucous glands to basically keep the airways moistened. And the hyaline cartilage, we can also see it contains 15 to about 20 C-shaped or horseshoe-shaped pieces of hyaline cartilage, and they are interconnected by the annular ligaments, which is dense connective tissue. And as you know, the cartilage basically helps prevent the trachea from collapsing and keeps the airways always like patent. We also have the tracheal muscle, which is a smooth muscle, and its contraction basically helps in decreasing the tracheal lumen, and it has the same function as I said. The reason why we do this is I explained before in the previous slide. It's basically just to keep moisture in and yeah, prevent it from drying out. We also have in the last part the adventitia, which is basically just loose connective tissue, contains lymphatic vessels and nerves. The adventitia also connects the trachea with other like components like the esophagus, the thyroid, and also the blood vessels, um, basically facilitating their stability and coordination like in their function. And on the right, you can see the schema of the trachea and the epithelium and the cross section. Here you can see a clearer view of how it is divided. So like I explained before, the cartilage is the most prominent. The mucosa is seen as well, the submucosa is seen, and the adventitia, which is located after the cartilage, is seen as well. And here we have the submucosa of the tracheal wall at a closer view. And over here, as you can see, the mucous glands are very clear. And the reason why this is because during the staining process, for example, with HE or anything else, we use an alcohol known as xylene, and that basically dissolves all the contents of the mucous gland, which basically presents it in the slides as a clear like slide. Um, and this is not applicable with the serous glands or serous acini. And yeah, we also have the serous demolune, as you can see here, at a clear view. And here's just more slides of the trachea. Yeah, these are just more slides of the trachea. Here's another slide in HE, but over here we can see that the elastic fiber is clearly highlighted. And on the last slide, it's just another slide of the trachea. And as you can see that the lymph node infiltration is there, again, to help it in monitoring any inhaled pathogens. And it also can secrete like cytokines to recruit more immune cells. Okay, that's it. Thank you for watching and see you next time.